PowerPoint presentation for Biology 104, Lab 9, Energy Production. The focus of this lab is on cellular respiration, and cellular respiration is the process by which cells use carbohydrates to produce ATP. Like the lab for photosynthesis, it's really important to know the overall equation. This is the overall equation for cellular respiration. Again, the main purpose is to take glucose and to generate ATP. In the equation, 1 glucose molecule plus 6 molecules of oxygen will produce waste product of carbon dioxide, waste product of water, and ATP. In a eukaryotic cell, cellular respiration occurs in the mitochondria. So that is the organelle, which is the site of cellular respiration. And this is a diagram of the mitochondria. This is the mitochondria. And cellular respiration involves 3 stages: glycolysis, the citric acid cycle, also known as the Kreb cycle, and the electron transport chain. And again, the main purpose of this is to take glucose and generate ATP. So all 3 stages generate some ATP. The overall reaction for cellular respiration. Again, this is the overall equation. 1 molecule of glucose, 6 molecules of oxygen produce 6 molecules of carbon dioxide, 6 molecules of water, and ATP. So what this process is doing is taking the glucose and breaking it down in a series of steps. And again, the main point is to generate ATP. ATP stands for adenosine triphosphate, and this is the molecule of usable energy for the cell. And remember from photosynthesis that oxidation is the loss of electrons, the loss of hydrogens, and in cellular respiration, glucose is being oxidized. Reduction is the opposite of oxidation. Reduction is the gain of electrons, the gain of hydrogens, and oxygen is being reduced in cellular respiration. The first stage in cellular respiration is called glycolysis. And glycolysis literally means glucose splitting. So in this process occurs in the cytoplasm. Glucose enters and 2-pyruvic acid exit. So when you split glucose, the result are 2-pyruvic acid molecules. Also during this process, ATP and NADH are produced. ATP is the readily usable energy. NADH is an electron carrier. So a diagram of this, start out with the glucose. It goes through the process of glycolysis, generating 2-pyruvic acid molecules and also producing some ATP, 2 molecules of ATP and 2 electron carriers. In order to go to the citric acid cycle, the pyruvic acid need to be prepared or prepped to go into the mitochondria, and this can be called the bridge or prep reaction. So for this reaction, 2-pyruvic acid enter. They are found in the cytoplasm, and they are converted into 2 molecules of acetyl-CoA, which will be able to enter the mitochondria. And in this process, another molecule of NADH is produced, so this is an electron carrier. And this is an example of the process. Pyruvic acid, pyruvate, goes through the process and is converted into acetyl-CoA, and 1 molecule of NADH is produced. The second stage in cellular respiration is the citric acid cycle, formerly known as the Kreb cycle. And this occurs in the mitochondrial matrix. It's the 2-acetyl-CoA enter this process. And as it goes through the process, it's going to produce ATP, readily usable energy, NADH, which is an electron carrier, and a second electron carrier called FADH2. And this is an example of the citric acid cycle. Acetyl-CoA goes in, it goes through the cycle, And as it does that, it's generating a little bit of ATP and lots of electron carriers, NADH, NADH, and FADH2. The last stage of cellular respiration involves the electron transport chain, and this is a structure that is found on the inner mitochondrial membrane. And this is where those electron carriers that have been generated during glycolysis, the prep reaction, and citric acid cycle, they drop off their electrons, and this is going to generate most of the ATP that is produced in cellular respiration. This is a diagram of the electron transport chain, which is found on the inner mitochondrial membrane. So this involves a series of proteins that are embedded in that membrane. And during this process, the electron carriers such as NADH and FADH2 will carry their electrons to this chain and drop the electron off to interact with those proteins, and these proteins will transport the electron along this chain, and in doing so, it will power the proton pumps to pump protons to the other side of the membrane. So this is active transport, transporting the protons to the other side of the membrane. Eventually, you build up a high concentration of hydrogen ions, protons on this side of the membrane, and eventually it'll be a high enough concentration that they will be allowed to diffuse through the ATP synthase. So they'll diffuse through the ATP synthase, and that will cause the ATP synthase to produce ATP. And also involved in this, for those electrons, as those electrons are transported along the electron transport chain, eventually they meet up with oxygen. So oxygen is required for this process to form water. As a review of cellular respiration, which is also sometimes referred to as aerobic ATP production. Aerobic because oxygen is required. And remember, oxygen was required at the very end of the electron transport chain. So as a review, it involves glycolysis, the Kreb cycle or citric acid cycle, and the electron transport chain. And ideally, going through this process, one glucose molecule can produce a maximum of 38 ATP. Another type of energy production is called anaerobic energy production. Ana means without, aero means oxygen. So this is a type of energy production that occurs when there is no oxygen present. We also know this process as fermentation. And fermentation is basically a type of glycolysis reaction. So the citric acid cycle and the electron transport chain are not involved. There are different types of fermentation. The types we are most familiar with are lactic acid fermentation and ethanol fermentation. And this produces a lot less energy than aerobic energy production. Out of one glucose, you get a maximum of only 2 ATP. And in aerobic energy production, cellular respiration, you could generate a maximum of 38 ATP. So ATP anaerobic production, this is an example of alcohol fermentation. So you start out with the glucose, it goes through glycolysis, produces the pyruvic acid, which will then be converted into ethanol. And also a byproduct of this is carbon dioxide. So you have carbon dioxide being produced. The other type of fermentation, lactic acid fermentation, again, start out with glucose, goes through the process of glycolysis, you end up with pyruvate, and now it gets converted into lactate. And in this process, no carbon dioxide is produced. The next part of this presentation is going to go over the experiments for Lab 9, and we'll show you what data you would have seen. The first part of your lab talks about a balloon demonstration, and in this demonstration, yeast would have been mixed with warm apple cider, and a balloon would have been put on top of the bottle. So what you would have seen is after 30 minutes, here is the bottle with the yeast and the cider mixture and the balloon on top. At 30 minutes, after 1 hour, this is what you would have seen, and these are the results you would have seen after 2 hours. So use these pictures as your observations to answer the questions in the lab report. For the next two experiments, the equipment you would have used are shown in the photo here. You would have used yeast, a suspension of yeast, glucose, a solution of glucose, and water. And then also you would have been using fermentation tubes. So these are all the fermentation tubes you would have been using. This is what a fermentation tube looks like up close. And fermentation tubes, of course, are used to determine the process of fermentation, how far it has progressed. For the fermentation tubes, you would have added all of your reagents, the yeast and the glucose and the water, if required. And they would have mixed together in the bowl, and then you would have tipped the tube so that the solution fills all the way to the top. So that's what you would have seen. If fermentation occurs, CO2 would have been produced, and the CO2 bubbles would have been collected in the top of the tube, and that would push the liquid down. So that is how a fermentation tube works. For experiment 1, part A, effective sugar concentration on fermentation, you would have set up 4 fermentation tubes with different amounts of 5% glucose, water, and yeast extract. And then again, you would have tilted them so that the solution was all the way at the top of that tube. Then these tubes would have been placed in a 37°C incubator for 60 minutes. And if fermentation is able to take place, you should see a bubble in these tubes pushing down the liquid. After 60 minutes, these are the results you should have seen. So this is tube 1, 2, 3, and 4. So in tube 1, there would have been no bubble. So the liquid is all the way to the top of the tube. The difference in color is these are the yeast cells that are just more dense than the water, and so they are falling down to the bottom. But here you would have seen a bubble. There's a meniscus here. Here, the bubble goes all the way down to that level, and here the bubble goes down to there. So the next part of the experiment would have been to measure the height of the bubble. Now for the data that you should record for experiment 1A. The first tube, tube number 1, had 0 height for a bubble. There was no bubble. This is tube number 2. That is the height of the bubble, so you should read the amount and record that on your lab report. This is the data for tube 3. Again, read that amount and record that on your lab. And this is tube number 4. And again, read the measurement of the height of the bubble and record that on your lab. The second experiment that you would have set up was experiment 1B, and this is the effect of temperature on the rate of fermentation. So for this, you would set up 3 more fermentation tubes in a similar manner. They would have had the 5% glucose, the water, and the yeast extract. And this time, the 3 tubes would be set at 3 different temperatures. So tube number 5 would have been put into the refrigerator, and the average temperature for refrigerators is 4°C. Tube number 6 would have been placed at room temperature. Average room temperature is about 20 degrees Celsius. And the third tube, tube number 7, would have been put in the incubator, which is at 37 degrees Celsius. And the results you would have seen after these tubes have been sitting for an hour would be tube number 5 would have no bubble, tube number 6 would have that bubble, and tube number 7 would have that size of a bubble. For recording your data for your lab report, tube number 5, you can use that to measure the bubble, virtually no bubble. Tube number 6, you can use that to measure the height of the bubble in tube number 6. And tube number 7, you can use that to measure the height of the bubble into tube number 7. The next experiment you would have done is experiment number 2, respiration rate over time. And for this, you are not using the fermentation tubes. Instead, you will be using test tubes. And each test tube represents a time point. So to set up this experiment, you would have taken your test tubes and in each test tube added 0.3% glucose and the yeast extract. You would have set up 6 test tubes and each one is a time point for every 10 minutes. When each test tube reaches the appropriate time point, you would have added Benedict's reagent, which tests for the presence of glucose. Then you would boil for 5 minutes and look for a color change. And remember, we've done the Benedict's before. You get a range of color. Blue means there is no glucose present, and that dark red means there is a high concentration of glucose present. So as you go up from blue to green to yellow to orange to red, you're increasing the concentration of glucose. So the data you would have seen if you had done this experiment would be here. 0 is the 0 time point. This is testing the amount of glucose that is present right when you set up the experiment. Then tube 1 is 10 minutes later from the initial time point. Tube 2 is 20 minutes later, 30 minutes later, 40 minutes later, and 50 minutes later. So record this data in your lab report and answer all the appropriate questions. The last experiment is experiment 3, testing aerobic cellular respiration activity. And for this, you would have looked at corn from 2 different groups, an A group and a B group. You would have taken 1 piece of corn from each group, cut it in half, and then exposed it to tetrazolium for about 15 minutes. And the results? This is what you would have seen. Group A, you would have seen a red color develop. Group B, no red color. So you can use those observations to answer the questions on the lab. So this should provide you all the data you need to complete your lab report.